So in 2026, the global pharmaceutical industry had a 97% failure rate for cancer drugs.
Yeah, 97%.
Like, let that number just sit with you for a second.
It's hard to even wrap your head around, honestly.
Right, you're talking about an industry wasting roughly $2 billion.
Hmm?
$2 billion. And 10 years of research to bring just a single drug to market.
And that's if they're lucky.
Exactly.
Mm-hmm.
Relying on this totally archaic system of trial and error chemistry that, I mean, it essentially boils down to throwing compounds at a wall and just, hoping something sticks.
Which is a terrifying way to do medicine when you really think about it.
It really is.
Hmm.
So today we are doing a deep dive into a sprawling, raw, and just incredibly dense set of documents about the woman who looked at that $2 billion failure rate and just decided to solve it.
Yeah, but not by inventing a new chemical. That's the crazy part.
Right, not a new chemical, but by building a machine capable of turning living, breathing human biology into a live, four-dimensional data stream.
Which is just wild.
We are diving into this story of Precigenetics, which is this boundary-pushing deep tech startup. Its founder, Parmita Mishra, and the fascinating internet ecosystem that kind of documented her rapid rise.
It really is an extraordinary set of source materials. We're primarily looking through the lens of this highly detailed blog post. It's titled Medicine, Precigenetics, Parmita Mishra, published by an author named Paramendra Kumar Bhagat.
Yeah, and this isn't just like some polished corporate profile, right?
Oh, not at all. The author has compiled biographical essays, raw technical product descriptions, and these completely unedited X threads.
Back when we still had to remind people it used to be Twitter.
Exactly. And he even includes his own, entrepreneurial pitches directed right at the founder. It gives us this perfect, unfiltered time capsule of March 2026.
Where high-level quantum optics collides with extremely raw startup survival and, internet hustle culture.
Yeah, the hustle is very real in this text, but the stakes could not be higher.
No, absolutely. If you're listening to this, I really want you to imagine holding a microscope. But instead of peering down at a piece of glass to see a dead, stained cell that's just frozen in time.
Which is how we usually think of microscopes.
Right. Instead of that, this microscope plays a live, continuous movie of the chemical reactions happening inside that cell in real time.
It's like switching from a photograph to a live stream.
Yes. It doesn't just show you the structure, it shows you the machinery of life operating second by second.
And our goal today is to figure out exactly how that works, why it hasn't been done before, and honestly, what kind of psychological profile it takes to build it.
Especially when everyone around you is calling it a literal delusion.
Oh, the pushback she got was insane.
We'll definitely get into that. But before we get into the quantum optics of it all, we kind of have to start with the fundamental communication problem.
Like translation issue.
Yeah. How do you even explain technology this dense to an investor, let alone the public?
Yeah.
The sources highlight this perfect exchange between the legendary tech investor Paul Graham and Parmita Mishra.
It's such a great interaction because it perfectly sets the stage for the engineering challenge ahead. So Paul Graham posted a thought on X, observing a recurring pattern he sees in Silicon Valley.
Okay, let's unpack this. What did he say?
He wrote, "Trying to make founders describe their products in simple everyday language is like trying to push together the north poles of two magnets."
Man, that is such a visceral image, right?
It really is.
You can almost feel that invisible physical resistance in your hands when you try to push magnets together.
Because you have these deep tech founders who spend 18 hours a day living in the weeds of molecular biology or physics.
Yeah, that's so deep in it.
And when a layman asks them what their company does, they inevitably hit you with a wall of jargon. They want to explain the intricacies of the lipid nanoparticles or the spectroscopy frequencies.
And the listener just immediately glazes over.
Exactly. The technical complexity actually repels human understanding.
But Graham is talking about that intense friction of translation.
Mm.
And Parmita Mishra replies to his post with a single 8-word sentence.
Just 8 words.
She writes, "Precigenetics turns living biology into computable data."
It's brilliant.
8 words. And later in the source material, she expands on that slightly, saying their mission is, quote, digitizing biology like we digitized intelligence.
That phrase is just, it's heavy.
I have been turning that phrase over in my head all morning. I mean, we have digitized text, we've digitized audio, we've digitized video.
Right, everything around us.
And with artificial intelligence, we've essentially digitized logic and reasoning. But biology has always been this chaotic outlier.
And if we connect this to the bigger picture, we really have to ask why. Why has biology historically resisted digitization?
Right. Why is it so hard?
Well, think about a line of computer code. It's deterministic. If A, then B. It's static. It's predictable.
It does what you tell it to do.
Exactly, but biology is the exact opposite. It's a wet, non-linear, constantly mutating environment. A single cell is an ecosystem of interconnected chemical reactions.
So if you poke one part of it.
Thousands of other variables shift in response. Historically, to digitize biology, scientists have had to reduce that complexity.
How do they do that?
They kill the cell, extract the DNA, sequence it, and turn that sequence into a text file.
Wow. Okay.
But a text file of DNA is just the blueprint. It is not the living, breathing building.
Right. It's the difference between reading the architectural plans for a house and actually watching people live inside it, open the doors, turn on the plumbing, and react to a fire.
That is a perfect analogy.
So Mishra is saying we are no longer just reading the blueprint. We are taking the fluid, chaotic, four-dimensional reality of the living cell and translating that continuous action into a structured binary code.
Code that a machine can actually read, and that is why her 8-word pitch is so powerful. It's a masterclass in strategic restraint.
Because she isn't just giving a slick elevator pitch.
No, she is clearly defining the massive, seemingly impossible physical engineering hurdle her company has to clear. To turn living biology into computable data, you cannot rely on software alone.
You actually need physical stuff.
You have to build a physical piece of hardware capable of capturing that immense complexity without destroying it. You have to build a bridge between the wet, chaotic world of the cell and the dry, ordered world of the silicon chip.
Which brings us to the actual hardware, the quote-unquote molecular microscope.
Right, the machine itself.
The source material describes Precigenetics as a microfluidic system integrated with advanced spectroscopy, drawing heavily on quantum optics.
That's a lot of big words.
Yeah, if I'm listening to this and I don't have a PhD in physics, my eyes are starting to glaze over a bit. Let's ground this. What is this machine actually doing?
Well, the text makes a huge point of emphasizing what the machine does not do.
Okay, like what?
It says no antibodies, no fluorescent labels, and crucially, no killing the cell to extract RNA or DNA.
Wait, here's where it gets really interesting, because I struggle with this. Scientists have been studying cells for decades, right?
Oh, absolutely.
We have incredibly powerful electron microscopes. We have massive genomic databases. Are you telling me that almost all of our high-resolution understanding of cellular reaction comes from looking at the equivalent of biological corpses?
To a very large extent, yes.
That's insane.
That last point you brought up, no killing the cell, is the absolute defining differentiator of this entire technology. To understand why this is revolutionary, you have to understand how traditional cellular biology currently operates.
Okay, walk me through it.
Right now, the gold standard in this space involves techniques like PerturbSec.
Let's define PerturbSec for a second, because that sounds like exactly the kind of jargon we just said we wanted to avoid. What is the actual mechanical process happening there?
So PerturbSec is a method where scientists introduce a perturbation.
Like poking the cell.
Basically, like a new drug or a genetic mutation. They introduce it into a cell, and then they sequence the RNA to see how the cell reacted.
Sounds reasonable.
But here is the critical flaw. To sequence that RNA, you have to destroy the cell. You literally break it open.
Oh boy.
You freeze the biological machinery in place, you extract the genetic material, and you take a static snapshot of its state at that exact millisecond.
But time is moving.
Exactly. We have learned an immense amount from those static snapshots, but we have fundamentally hit a ceiling. Biology is entirely dictated by the temporal dimension. Time is everything.
Because it's a living system.
Right. It evolves second by second. When you introduce a drug to a cancer cell, it doesn't just instantly die or instantly heal.
It's a process.
The drug binds to a receptor, which triggers a protein, which signals the nucleus, which begins altering gene expression over the course of minutes, hours, or days.
Yeah.
If you take a static post-mortem snapshot, you're only looking at the aftermath.
It's the difference between a security camera and a crime scene photo.
Yes. Think about the mechanics of a crime scene photo. You arrive at a bank after a robbery. You take a high-resolution photograph of a broken window, an open vault, and some scattered cash on the floor.
You can figure out some stuff from that.
Sure. You know the glass was broken from the outside. You know the vault was breached.
But you have no idea how they did it.
Precisely. You don't know if they used a crowbar or a laser cutter to get into the vault. You don't know if it took them 5 minutes or 5 hours.
Right.
You don't know how many people were involved or what order they moved through the room. Now imagine having a high-definition security camera that recorded the entire event from the moment they approached the building to the moment they fled.
You see the whole story?
That camera shows you the mechanisms of the event over time. In biology, if you have to destroy the cell to study it, you lose the very movie you were trying to understand. You are forever stuck guessing what happened between snapshot A and snapshot B.
Okay, that analogy makes the problem incredibly clear. But it also raises a massive question about the solution for me.
Which is,
If we aren't breaking the cell open and we aren't using those fluorescent dyes to make the molecules glow in the dark, how on earth is this machine actually seeing what is happening inside the nucleus?
it's basically magic, well, physics.
Are we just guessing based on how the outside of the cell behaves?
Mhm.
The text mentions label-free optical spectroscopy. Break down the physics of that for me. How do you look inside a living thing without altering it?
It relies on the fundamental principles of how light interacts with matter.
Okay.
When you use fluorescent labels, which is the standard way biologists highlight specific proteins, you are chemically attaching a glowing tag to a molecule.
To make it light up under a microscope.
Right. But molecules in a cell are incredibly small and precisely balanced. When you attach a massive glowing tag to a protein, you are altering its weight, its shape, and potentially its natural behavior.
Oh, I see. It's like putting a 50-pound backpack on an Olympic sprinter and then trying to study their natural running form.
Exactly.
The act of observing them has fundamentally ruined the data.
Yeah. You are no longer observing natural biology. You are observing altered biology. Precigenetics avoids this entirely by using advanced spectroscopy.
So no backpacks.
No backpacks. Every type of molecule, whether it's a lipid, a protein, or a nucleic acid, has a specific unique vibrational signature. Think of it like a tuning fork.
Okay, I'm with you.
If you shine a very specific, highly controlled laser light, and this is where the quantum optics comes in onto a living cell, the light interacts with the chemical bonds of those molecules.
Does it damage them?
No, most of the light passes through or scatters normally, but a tiny fraction of the light actually changes its energy level based on the vibrational frequency of the molecules it hits.
So the light bounces back slightly changed, and the machine reads that change.
Yes. By capturing and analyzing that scattered light, the spectral fingerprint, the machine can identify exactly which molecules are present, what shape they are in, and how they are shifting.
All without touching it.
Without ever touching the cell chemically. It is entirely non-invasive. The cell has no idea it is being observed.
That is staggering. The sources say this allows them to read lipid content, protein conformation, and capture live videos of epigenetic changes inside the nucleus.
Which is the holy grail.
And just to define epigenetic changes, for a moment, we aren't talking about the DNA sequence itself changing, right?
Right, your DNA stays the same.
We are talking about the software of the cell, the mechanisms that turn specific genes on or off in response to an environment.
Yes, exactly.
So Precigenetics is watching the cell rewrite its own software in real time as it fights off a disease or reacts to a chemical.
And they are capturing it in 4 dimensions, 3 spatial dimensions plus time. It is an unprecedented level of biological observation.
So let's look at this from a purely business perspective.
All right.
You have successfully engineered what might be the most revolutionary diagnostic tool in the history of modern biology.
Arguably, yes.
You have a microscope that can watch the secrets of life unfold without killing the subject. If I am an investor, or even just a pragmatic founder, the natural next step is incredibly obvious.
You'd think so.
You patent the hardware, you box it up, you slap a multi-million-dollar price tag on it, and you sell it to every major hospital, research university, and clinical diagnostic lab on the planet.
You become the MRI machine of the 21st century.
Exactly. But according to the sources, Parmita Mishra did something totally counterintuitive.
What's fascinating here is she actively rejected that path.
Really?
The text reveals that she spent 4 agonizing months deliberating on this exact strategic choice. The pressure to go into clinical diagnostics must have been immense.
I mean, diagnostics represent immediate, proven cash flow. If you have a machine that can tell a doctor exactly how a patient's live cancer cells are reacting to chemotherapy, hospitals will pay almost anything for it.
But she walked away from the immediate money.
And I want to dig into why. Because on the surface, refusing to sell your groundbreaking invention sounds like terrible business.
It sounds like startup suicide.
Right. But when you actually look at the friction involved in the medical device industry, it starts to make sense. Going into clinical diagnostics doesn't just mean selling a machine. It means entering a bureaucratic nightmare.
It means dealing with the FDA for years before you see a dime of revenue. It means fighting with insurance companies over CPT codes and reimbursement rates.
Oh, the billing codes.
Exactly. It means building a massive global sales force, managing supply chains for hospitals, dealing with maintenance contracts, and ensuring absolute regulatory compliance in a dozen different countries.
Right. And suddenly, your cutting-edge quantum optics startup isn't a science company anymore. It's a medical compliance and distribution company.
You lose the plot.
You have diverted 90% of your energy away from pushing the frontier of physics and poured it into arguing with an insurance claims adjuster in Ohio about whether your cell scan qualifies for a specific billing code.
Which is why this decision is a masterclass in long-term strategic restraint. Mishra recognized that selling the machines would dilute their ultimate focus.
So what did she do?
Instead, she chose to keep the hardware in-house. She focused on building expensive, room-sized, immensely complex instrumentation that stays at the absolute frontier of research.
So she doesn't care about making them small.
Right. They aren't constrained by what is commercially viable to ship in a box. They can continually upgrade the lasers, the microfluidics, and the compute power without worrying about whether it will fit through a hospital doorway.
But the risk there is astronomical. I mean, if you aren't selling the machines, how do you make money?
That's the million-dollar question, or billion-dollar question.
You are relying entirely on venture capital to keep the lights on. You are betting the entire future of the company on the idea that the data these machines generate will eventually be worth exponentially more than the physical machines themselves.
And that brings us to the core economic engine of Precigenetics, which the text refers to as the data flywheel.
Okay, the data flywheel.
This is the key to their entire valuation. By keeping these super-resolution machines in-house or deploying them only within very tight exclusive research partnerships, Precigenetics owns the generation engine.
So they own all the output.
Every single time they run an experiment, every time they capture a live movie of a cell reacting to a drug, they add to a proprietary database.
And because they're the only ones with this specific label-free technology, they're the only ones generating this specific kind of data.
Exactly. Every improvement in their sensor technology compounds their data advantage. They aren't trying to be the company that sells the camera.
Or they're being the movie studio.
They are building the world's highest resolution, most exclusive film studio, and they are retaining the copyright to every single frame of footage they shoot. As that database grows, it becomes an insurmountable moat. No competitor can catch up because no competitor has the years of historical 4D cellular footage that Precigenetics has already archived.
Okay, so they have built this massive proprietary vault of live cellular movies, but who is the audience for this footage?
Good question.
It's not human doctors sitting in a dark room watching hours of a liver cell vibrating.
Yeah.
The sheer volume of information is way beyond human comprehension.
Far beyond it.
This leads us directly to the ultimate mission of the company. The consumer of this data flywheel isn't human at all. It is artificial intelligence. Mishra is building the eyes that AI needs to finally solve human disease.
To understand the magnitude of this, we have to look back at the broken pharmaceutical pipeline we mentioned at the start. Why does it cost $2 billion in 10 years to make a drug, only to see 97% of oncology candidates fail?
Because it's guesswork.
The text argues that the system is broken because AI currently lacks a comprehensive, high-quality data set of live drug cell interactions.
I mean, if you think about how we train artificial intelligence today, it is entirely dependent on the quality and volume of the training data.
Garbage in, garbage out.
Exactly.
Yeah.
You want an AI to write code, you feed it millions of repositories from GitHub. If you want an AI to generate images, you feed it billions of captioned photographs from the internet. But if you want an AI to predict exactly how a novel chemical compound will affect a human pancreatic cancer cell over 48 hours, where do you get the training data?
It doesn't exist. Or rather, it didn't exist before Precigenetics.
Wow.
The text makes a brilliant comparison here to AlphaFold.
Oh, AlphaFold. That was huge.
AlphaFold was a massive breakthrough by Google DeepMind that essentially solved the protein folding problem. It could look at a sequence of amino acids and accurately predict the complex three-dimensional shape that protein would fold into.
It was hailed as one of the greatest scientific achievements of the decade.
But the author of this blog post makes a really crucial point about AlphaFold. AlphaFold didn't generate its own biological data. It was purely a computational achievement.
Wait, really? Where did it get the data?
It only worked because human scientists had already spent decades painstakingly mapping the static 3D structures of hundreds of thousands of proteins using X-ray crystallography and cryo-electron microscopy.
Oh, so humans did the heavy lifting for the data collection.
Right. They had built a massive public library called the Protein Data Bank. DeepMind took that existing library, fed it into their neural networks, and trained the AI.
So AlphaFold stood on the shoulders of decades of static structural data.
Exactly.
But there is no equivalent public library for dynamic functional cellular responses. Humanity has never possessed a comprehensive temporal data set showing how living cells react to millions of different perturbations over time.
Because we always killed the cell.
Exactly. So Precigenetics is building that missing library from scratch. They are generating the data that will allow the next iteration of AI to move past static structure and move into dynamic prediction.
And this raises an important question. What happens when we get there? The ultimate goal of all of this is what Mishra calls rational drug discovery.
I really want to linger on that phrase because it is the antithesis of how medicine works today.
Oh.
Rational drug discovery means we stop guessing. It means moving away from the brute force method of let's synthesize 10,000 random chemicals, test them on mice, see which mice survive, and then hope it translates to humans.
Which, as the 97% failure rate shows, it rarely does. Mice are not humans.
Right. Rational drug discovery means we ask the AI, design a molecule that will specifically alter the epigenetic state of this exact type of human lung cancer cell without damaging the healthy tissue around it.
And the AI can actually do it.
The AI consults its vast database of live cellular footage, simulates the exact chemical interaction down to the subcellular level, and outputs the precise molecular structure required. We predict the cellular outcome before the drug ever touches a physical patient.
We design cures based on observed computational reality, not statistical luck.
And when you start looking at the sheer scale of the math involved in that kind of simulation, it is truly staggering. The source material breaks it down.
It's mind-bending.
The average human body contains roughly 40 trillion cells. Inside every single one of those cells, there are approximately 1 trillion chemical reactions happening every single second.
40 trillion cells. 1 trillion reactions per second per cell.
If you are listening to this, just try to wrap your mind around the sheer volume of chaotic activity happening inside your own body right now, just to keep you sitting upright and breathing.
It's a miracle we function at all.
You cannot put a human mind to the task of mapping that. You can't even put a room full of the smartest PhDs in the world to that task. The complexity is beyond biological comprehension. You need massive civilization-scale compute.
But as the saying goes, compute without high-quality data is useless. You can have the biggest supercomputer on Earth, but if you feed it those static post-mortem snapshots of dead cells, it will output flawed predictions.
Because it's learning from a crime scene photo.
Right. Mishra's core philosophy is that once you feed an AI a continuous stream of label-free, site-specific data from living cells, the entire paradigm of medicine completely changes. Disease is no longer a late-stage descriptive label.
Right, you don't just say, oh, the patient has stage 3 cancer.
Exactly. That is a macroscopic observation of a failure that began months or years earlier at the molecular level. With this technology, disease becomes a fully mapped epigenetic progression. It is a sequence of events that can be tracked, predicted, and ultimately intercepted before it ever manifests as a macroscopic symptom.
I want to act as a proxy for the listener for a second here. If you were listening to this on your commute or while washing dishes, I want you to pause and reflect on a time when you, or maybe a loved one, had to navigate a serious illness that relied on trial and error dosing.
It's an awful experience.
Think about the reality of treating severe depression, or autoimmune disorders, or undergoing chemotherapy. You sit in a doctor's office and they look at you and say, well, the standard protocol is to try this dosage of this compound for 6 weeks. Let's see how the side effects are. Let's see if the tumor shrinks. If it doesn't work, we'll pivot and try a different cocktail.
It's physically brutal.
It is an exhausting, terrifying, physically brutal process. You are essentially acting as a human guinea pig for a highly educated guess. This technology, this combination of quantum optics and artificial intelligence, aims to eradicate that medical guesswork entirely.
It is an incredibly ambitious mission, perhaps one of the most ambitious technological pursuits of the decade. And to take on an entrenched, multi-trillion-dollar industry that is comfortable with a 97% failure rate requires an equally formidable architect.
Oh, for sure.
You don't attempt to digitize human biology unless you have a very specific, almost unreasonable constitution. Which brings us to the relentless builder behind Precigenetics, Parmita Mishra.
I am so fascinated by her origin story because it completely recontextualizes her ambition. When you look at her background, you realize she isn't just a Silicon Valley tech bro trying to disrupt an industry for a quick payout.
Not even close.
She comes from this profound, deeply visceral legacy of healing in India. The text states that her parents are both physicians who, collectively, have saved over 50,000 lives.
50,000 human beings are walking the earth because of her parents.
That is a staggering legacy to inherit. And the source material goes into specific detail about her mother, Dr. Parmita Mishra.
She sounds like a superhero.
She really does. She is a pediatric cardiologist. The text describes how she travels across India performing surgeries to close holes in children's hearts, often doing it completely for free for families who cannot afford it.
And she founded an NGO too, right?
Yeah, a non-governmental organization to support underprivileged children facing cardiac issues. Parmita describes her mother coming home from these grueling hospital shifts, staying awake until 2 or 4 in the morning just to spend quality time with her daughter and teaching her what it means to be a fighter.
There is this deeply moving tribute that Parmita posted on X on Women's Day in 2026. She calls her mother the smartest woman on earth and openly declares, "I live to make you proud, Mama."
It's such raw emotional fuel. When you look at Parmita's resume, you see the elite academic pedigree. She went to an Ivy League university, she studied computational biology, she published peer-reviewed research, she gave invited talks at MIT and Berkeley, she started her own lab.
She has the ultimate academic credentials.
But that pristine, high-level academic brilliance is fused with this intense, gritty, frontline medical legacy.
If we synthesize those two halves of her background, the Ivy League computational theory and the frontline pediatric cardiology, it explains her entire approach to Precigenetics.
How so?
Her academic journey actually exposed the blind spots of traditional biology to her. She didn't just passively study the field. She probed its limits. She saw the diagnostic muddy waters we discussed earlier, the reliance on dead cells and fluorescent dyes, and she grew intensely frustrated by them.
Because it wasn't solving the real problem.
Exactly. She realized the academic tools she was being handed were insufficient to solve the problems her mother was facing in the operating room.
And think about the psychological armor you develop when you grow up in that environment. I always find it fascinating how different founders process risk.
Risk tolerance is huge in startups.
Right. In Silicon Valley, there is this pervasive fear of failure. Founders are terrified of losing a seed round or having a product launch flop on Product Hunt. But when you grow up watching your mother sacrifice her own sleep to perform free open-heart surgery on an impoverished child, it completely inoculates you against the superficial fears of the tech industry.
Absolutely. What is a failed venture capital pitch compared to a child dying on an operating table?
It gives her this fearless intensity because her baseline for what constitutes a real crisis is completely different.
That fearless intensity is exactly what was required for the next chapter of her story, because as the text explicitly frames it, launching Precigenetics wasn't viewed as a bold scientific endeavor by those around her. It was viewed as a leap into pure delusion.
Let's walk through the timeline of this so-called delusion because it is wild. The text notes that she incorporated Precigenetics on May 5, 2024.
Okay.
She started the company with exactly $1,000 and a personal loan from a loved one. She had no massive war chest, no institutional backing at that moment. In a very raw, unfiltered thread from March 2026, she reflects on those early days and writes, "I gave away everything for Precigenetics, everything."
The social and personal fallout she describes from that decision is severe. She writes that her parents were rightly not happy about her throwing away a stable academic career.
Which is understandable.
Sure, her ex-partner was appalled, and perhaps most cuttingly, her own childhood friends explicitly told her she needed to visit an asylum.
Wow. I cannot imagine the isolation of that moment. You have the vision for the future of human health in your head, and the people closest to you think you have literally lost your mind.
But her response to the isolation is incredible. She writes a post that effectively becomes her personal manifesto.
What does she say?
She says, "Not all crazy people belong in an asylum. Some of them bring their delusion into the tangible realms. through the sheer act of building persistently.
Through the sheer act of building persistently. It is a profound statement on the nature of innovation. Society often defines insanity as seeing things that aren't there.
Right.
But visionaries also see things that aren't there, they just haven't built them yet. The only difference between a delusion and a breakthrough is execution.
And the execution here is whiplash-inducing. I struggle to see how a startup with $1,000 in a loan builds a room-sized quantum optics machine in a year. Walk me through the actual tangible milestones, because it sounds impossible on that budget.
It speaks to her sheer force of will and ability to attract the right believers early on. Despite the personal pushback, she managed to secure pre-seed funding from the 1517 Fund, which is known for backing deep tech outliers.
Okay, so that gets them some runway.
Yeah, and she managed to get a Nobel laureate on her advisory board, giving her immense scientific credibility. The text also mentions a crucial 5-minute phone call with Gary Tan, the CEO of Y Combinator, during a particularly low moment that helped reassure her path.
Those connections are huge.
And by late 2024, just months after incorporating, they had built Pro-T01, their first functional hardware prototype.
Just months. That's insane for hardware.
And the momentum just accelerates from there. By May 2025, exactly 1 year after starting with that $1,000, she has shipped a custom hardware stack that cost over $500,000 to build.
Wow.
She gets accepted into the highly competitive NBC Biolabs incubator space. And most importantly, the machine actually works. She is generating unprecedented super-resolution data of live cells that nobody else in the world has.
The contrast between the societal accusation of insanity and the undeniable physical reality of $500,000 of custom hardware generating novel scientific data is stark. It proves her manifesto true.
But what stands out to me in the source material isn't just the timeline of success, it's the incredible vulnerability with which she shares it.
Yeah, she doesn't hide the hard parts.
If you look at most deep tech hardware companies, their public communication is heavily sanitized. It is entirely controlled by PR firms. You get highly polished press releases about funding rounds and sterile photos of clean rooms.
Very corporate.
But Parmita is on X, bleeding on the timeline. She is openly discussing her lack of sleep, her emotional state, and the personal cost of the company.
That vulnerability highlights a massive systemic issue that the source material tackles head-on. The friction she faced wasn't solely about the technical ambition of a label-free microscope. She explicitly outlines the unique and deeply entrenched bias she faces as a female founder building capital-intensive hardware in a male-dominated deep tech space.
Let's read her direct quotes from the text, because she does not mince words about this reality.
Yeah, let's hear it.
She writes, quote, "People doubt women. People call me crazy. People call me dangerous because I have emotions for my job, and men will never be told what I'm told."
If we unpack that claim that men will never be told what I'm told, we have to look at the historical context of venture capital and hardware engineering.
So what does this all mean for her?
Well, deep tech hardware is exceptionally capital intensive. You are asking investors for millions of dollars before you ever have a product to sell. In that environment, investors often pattern match for a very specific type of stoic, detached, usually male founder.
The classic Silicon Valley archetype.
Exactly. When a male founder exhibits intense, obsessive passion for his hardware, he is often lauded as a visionary or driven genius. But when a female founder exhibits that exact same intense emotional connection to her life's work,
she is labeled dangerous. She is called crazy or unhinged. Her passion is weaponized against her as evidence of instability rather than dedication. It's a wildly frustrating double standard.
it really is.
If a guy sleeps under his desk to build a rocket, he's on the cover of a magazine. If she sacrifices everything to cure cancer, her friends tell her to check into an asylum.
Exactly. And what is fascinating is how she chooses to process that systemic friction. She doesn't write a sanitized op-ed complaining about it. She completely leans into the labels, almost weaponizing them for her own momentum.
Oh, her response is amazing.
She follows up those observations by stating, quote, "I am dangerously unhinged and crazy and a woman, and it is why I will make it happen."
The absolute defiance in that sentence is breathtaking. I am dangerously unhinged and crazy and a woman, and it is why I will make it happen.
It's a mic drop.
She is not asking for permission to be in the room. She is not modulating her tone to make older male investors feel more comfortable. She refuses to apologize for having emotions about technology that is designed to save human lives. She is essentially demanding to be judged solely by the data output of her machines.
She articulates this broader philosophy in another quote from the text. She says, "Brilliance is never easy to come around, and Precigenetics is brilliant, and it is brilliant because it does not care for sanity. It cares for science."
She is drawing a very stark line in the sand there between societal expectations and scientific reality. If you want to be viewed as sane by polite society, you build another sauce app for middle management. You take the easy route.
Yeah, you build an app.
You build a diagnostic tool, get your insurance billing codes, and make a safe exit. But curing cancer, mapping the 4D biology of a human being, rewriting the fundamentals of rational drug discovery, that requires a level of obsession and disregard for norms that society will always code as insanity. And she's clearly made peace with that label.
She has embraced the fact that paradigm-shifting science requires an almost unreasonable level of conviction.
Now, to fully appreciate how this entire narrative, the quantum optics, the AI data flywheel, the deeply personal origin story, and the raw internet defiance how all of this actually reaches us, we have to zoom out and look at the framing of the source material itself.
The blogger.
Yes, because we aren't reading an academic journal. We're getting all of this insight through the specific lens of a blog post written by Parmita Bhagat. And examining the ecosystem surrounding this post is a fascinating journey into the digital culture of 2026.
It is a perfect micro-study of the modern internet. When you look closely at the text, you realize that Paramendra isn't acting as a detached, objective journalist observing a subject from afar. He is actively participating in the narrative.
He's right in the middle of it.
The blog post documents his real-time hustle on X. He sees Parmita posting about her technology, and he jumps into her replies to pitch himself.
It's incredible. He literally replies to the CEO of this bleeding-edge deep tech startup and says, "You should also go in the other direction. Write an X article and write a book. Can hire me as a ghostwriter."
Just straight up asking for a job.
He pitches her a flat $5,000 gig to write a book explaining her microfluidic quantum optics in simple terms, comparing the proposed tone to his own previously published book, How Big is the Universe? He doesn't stop there.
He keeps going.
He also pitches launching a highly produced, well-edited weekly web show for the company to help them with PR, recruiting engineers, and fundraising. He is shooting his shot right there in the public square.
And while on the surface that might seem purely opportunistic, if you analyze the needs of a company like Precigenetics, his pitch actually makes a tremendous amount of sense. It brings us all the way back to the Paul Graham quote about the magnets.
Deep tech has a massive communication deficit.
Exactly. Deep tech needs translators.
That is such a crucial point. If you are Parmita Mishra, your primary job is to ensure the microfluidics don't leak and the lasers are calibrated to the exact sub-nanometer frequency. You do not have the time to write an accessible narrative for the general public.
No, you're in the lab.
But you desperately need the public, and more importantly, massive institutional investors to understand your vision.
Exactly. To raise the billions of dollars required to map human biology and train biological superintelligence, you cannot just hand an investor a densely packed, peer-reviewed paper on label-free optical spectroscopy. They won't read it.
They'll fall asleep.
You need a compelling, digestible narrative. You need the story of the pediatric cardiologist's mother, the 97% failure rate, and the 4D molecular movie. Paramendra recognizes that she has the world-changing science, but she needs the bridge to the public. He's offering to build that bridge.
And when you look at the sheer volume and variety of his blog sidebars included in the source material, it paints this wild, vibrant picture of the digital zeitgeist in early 2026.
It's quite the list of topics.
This is a guy who is writing sci-fi novels about a concept called calquism. He's posting dense essays about agentic AI, he's tracking crypto markets, he's writing about tech incubators, and then pivoting to geopolitical musings on the Middle East and the defense of Taiwan.
He is incredibly prolific.
And his interests are scattered across every major frontier of human development.
His blog is a reflection of an internet ecosystem that is entirely obsessed with the concept of impending abundance. He is chronicling a moment in time where artificial intelligence is rapidly advancing and people are waking up to the realization that AI isn't just going to write emails for us, it is going to physically alter our reality by converging with fields like biology, energy, and material science.
We even see mentions in the text of the broader YouTube media ecosystem that is starting to surround her. There are titles of videos like The Biotech Founder Reading Your Genes Like Code, and long-form podcast deep dives called How to Think About Biology.
She's becoming a recognizable figure.
It shows that Parmita isn't just building a physical machine in a basement laboratory in isolation. By operating so openly, by bleeding on the timeline, she has become a central protagonist in a much larger cultural conversation about the future of human health and the capabilities of AI.
She represents the physical manifestation of the Internet's highest hopes for technology.
Which brings us to the end of this truly incredible journey. Let's distill everything we've unpacked today. We started with a cryptic tweet about magnets and the resistance of technical translation.
Right.
From there, we unpacked a multi-million-dollar optical microscope that uses the vibrational signatures of light to keep human cells alive while recording real-time, four-dimensional movies of their most intimate chemical reactions.
We explored the deeply personal motivations of a founder honoring her mother's frontline medical legacy. We analyzed her strategic brilliance in rejecting the easy, immediate money of clinical diagnostics in order to build an insurmountable data flywheel.
Yeah, that pivot was everything.
And we looked at the sheer grit required to build complex hardware in public while facing down intense systemic skepticism and accusations of delusion.
And the ultimate synthesis of all of this, the reason this specific startup matters in the grand scheme of things, is that Precigenetics represents a monumental paradigm shift. It's the shift from treating human biology as a dark, unreadable, chaotic mystery that we can only guess at, to solving it as a deterministic engineering problem driven by massive compute power and continuous live data.
The next time you are scrolling the news and you read a headline about a highly anticipated miracle drug failing in late-stage phase 3 clinical trials after a decade of research and billions of dollars spent, remember Parmita Mishra's mission.
It really puts it into perspective.
She is building the eyes to ensure that an artificial intelligence can predict that exact molecular failure before a single human patient ever has to endure the false hope of a doomed trial.
It changes the entire landscape of medicine. And as we wrap up, it leaves me with a final, lingering thought for you to mull over as you go about the rest of your day. Think back to our opening discussion about the 97% failure rate and the terrifying medical guesswork of trial and error dosing.
It's a sobering thought.
If Precigenetics actually succeeds in their mission, if human biology truly becomes as computable, as readable, and as predictable as a line of software code, how does that fundamentally change our psychological relationship with our own mortality?
Wow.
If an artificial superintelligence can perfectly simulate your unique body's response to any compound in real time down to the subcellular epigenetic level, is illness no longer a tragic, unavoidable twist of fate? Is a cancer diagnosis no longer a death sentence, but simply a line of buggy code waiting to be rationally debugged by a machine?